So this is Nursing 2533, Process of Human Diseases 1, Unit 9. So we're going to talk about developmental and genetic disorders. Okay. We need to start out with some definitions. A lot of times words are used that have precise meaning, but we use them with kind of more generic, widespread meaning. And in a study like this, we can't. So we have to lose some of the lay meanings. So when we talk about congenital disorders, we're talking about disorders that are present at birth. Now, that doesn't include the disorders where there's a genetic factor that is there at birth and doesn't appear till later. That wouldn't be considered genetic, or that wouldn't be considered congenital. It would be considered genetic. Now, there's a couple of different ideas that need to be addressed here. Most of us have some recessive genes. Most of us have genes that are not fully functional, or that will lead to some difference in physiology, but they're not expressed. They don't really matter, except when we pass them on to our children. And if our children get 2 copies of that recessive gene, then we can end up with a disorder. So most of us are carriers of some sort of recessive gene. So there are these genetic things that can contribute to congenital disorders, but there's also environmental factors. So this is exposure to various different different agents that can cause birth defects and that kind of thing, and whatever so usually there's some sort of combination, or often there's some sort of combination between genetics and environment, but environmental factors are really important. Now, I've already said congenital disorders are obvious at birth. They're present. Now, they can have a genetic or an environmental cause, or both. Congenital malformations are when parts get built wrong. So some of the like some of the heart defects we talked about, these are structural defects, and half of them are genetic, but that means the other half are environmental. So the teratogens are the environmental factors that can affect the developing fetus. Now, there is a problem, and that is that most of the organ systems, most of the fundamental structure of the systems happen in the first 8 weeks of fetal development, and oftentimes people don't even know that they're pregnant until near the end of the first trimester. So they can be exposed to a lot of these factors when they don't even know that they're pregnant. And these factors that the mother gets exposed to can affect the initial development of an organ system, and therefore that system will not develop correctly. That's really one of the reasons why it's very important that if you are planning pregnancy, to be very careful about teratogens. So, like I said, some inherited disorders aren't obvious at birth. They don't come up till later. Like things like Huntington's chorea doesn't come on till the fourth decade of life. But you have it right from birth. It's just it doesn't express. With advances in genetics and being able to look at DNA and having mapped the human genome, we can find these things in people and prepare them for it. In the past, it was really a crapshoot. When they were going to show up. So a couple of other definitions. Phenotype versus genotype. Phenotype is the expression of your genes. This is the physical characteristics of a person. So your phenotype is that you have blue eyes. It's which genes are expressed. Blonde hair, just the shape of your nose, the development of your body, the type of collagen you make, all kinds of things are the phenotype. So these are the genes that are expressed, and it is the physical nature of your body. The genotype is really what genes do you have, not really the ones that are expressed. So the genotype is the genetic makeup of the person. We have 23 pairs of chromosomes, and each one of those is made up of a whole bunch of genes. Now, if we think back to AMP, we talked a fair amount about this and about what chromosomes look like. Now, in interphase, most of the time in the cell, the nucleus, the genetic material is found as chromatin, and it's only during meiosis or mitosis that it condenses down into chromosomes that are easily seen. They look like an X. In the middle of it, there is a centromere that holds the two arms of the X together. Each arm is identical, and we call those things chromatids. So two chromatids are joined by a centromere to make a chromosome. We talked about haploid and diploid numbers back then. So the diploid is the number of chromosomes you have in pairs, right? The haploid number is the number of pairs that you have. So in humans, the diploid number is 46. The haploid number is 23. Now, in each pair, you have one from your mother and one from your father. Which one gets expressed depends on the genes themselves. We call 22 of the 23 pairs the autosomal chromosomes. These are not the sex-linked chromosomes. They're all the rest of them. The sex chromosomes really are the last pair. Now, they're called that because they determine the sex of the individual. And that's the X and Y chromosome. So, an allele is the two copies of the gene located on the same site on each chromosome. So, say your chromosome 12, there's a gene on it that you got from your mother, and for eye color or whatever, I don't know which chromosome actually has eye color, but say on a chromosome pair, there's a gene for eye color. You got the one gene from your mother. You have the one gene on the other chromosome of that pair from your father. Those two chromosomes are called alleles. So say you have a chromosome for blue eyes from your mother, and the allele is brown eyes from your father. The brown-eyed allele will dominate over the blue-eyed, and you would have brown eyes. Although you would have a recessive allele for the blue, which isn't expressed. So the dominant alleles are the ones that express into the phenotype. The recessive alleles are the ones that you just carry, and the trait isn't expressed. But you can pass on that allele to your kids. So, and it may get expressed in a generation or two or three. Like it can, it kind of moves down the chain that way. Punnett squares. Punnett squares are the looking at the relationship between the dominant and recessive alleles, and I'll talk about them when we talk about the single gene disorders. So when we look at a chromosome, they end up looking like this. Is a photomicrograph of it. You can see that it's egg-shaped. The centromere is here in the center. That arm and that arm are identical. This is where it splits during anaphase and telophase to create the daughter cells in mitosis. But I'm not going to review that now. Okay, more definitions. Multifactorial or polygenic disorders mean that just what it sounds like. There's many factors contributing to the disorder, that it's not just a simple single gene screw up that causes it. There has to be a lot of genes involved, or genes and environment. We tend to call this hereditary disorders, the things that run in families. But they need more than one factor. There's an old saying in pathology that says that genetics loads the gun, but environment pulls the trigger on a lot of these things. So this could be things like rheumatoid arthritis, etc. Okay, the term mutation means that the genetic material gets screwed up somehow, and that it becomes a permanent change in the DNA as the DNA gets reproduced during the S part of the cell cycle. Now, these things often, like, we are exposed to mutagens all the time. Just the radiation that comes off your computer screen, that comes off your cell phone, has mutagenic properties. The X-rays, different environmental factors, solvents and different pollutants can have these mutagenic factors. But generally speaking, we are pretty good at not having too many mutations. And oftentimes, if a mutation is really bad, then that cell doesn't survive, and and therefore doesn't pass it along. So they kind of sort themselves out for the most part. But some of them persist, and some of them become new genetic information that can be passed along and cause disorders. Okay, another definition, genetic disorders. So genetic disorders are disorders that are present at birth or appear later in life, but they're inherited for the most part. So a genetic disorder may be congenital or it may not be congenital. A genetic disorder you get from your parents. Now, sometimes, and I think this number is a little bit high, 15 to 20 is what it says on this slide. I learned kind of 10 to 15%, but it's in that neighborhood. Genetic disorders are novel. They appear during fetal development due to mutation. Now, there is a school of thought that says that all genetic disorders initially started as a genetic mutation that happened during fetal development that was compatible enough with life that it became an inheritable disorder. There's a disorder that is a genetic disorder that we see a fair amount down around the Windsor area. It's called malignant hyperthermia. I won't get into what it does, but it basically is that these people, when they spike a fever, can't break the fever. It's a very dangerous disorder. And there's kind of historically, there's two pockets of this where it is not as rare as it is everywhere else. One is in central France, and the other one is in the Windsor area. And most of the people that have it are of French-Canadian descent, which means that it probably goes back to somebody who emigrated from France in the 1700s. Now, from what I understand, they've done the research, and this disorder can all be traced back to one individual living in the 1600s in France who had a mutation that screwed up the way that the pyrogens act, and it's completely compatible with life, and so all his offspring, multi-generational, potentially have this disorder. But the point is that it started as a genetic mutation in one individual and has become a genetic disorder, an inheritable genetic disorder, in thousands and thousands of people. So it's a bit of a gray line. Okay, so when we talk about genetic disorders, we're going to talk about four groups of disorders. So the first ones we're going to talk about are the chromosomal abnormalities, and this is screw-ups with meiosis and mitosis. The second one we're going to talk about is single gene disorders, where the disorder is caused by a single recessive gene. The third type are going to be the sex-linked disorders. And the fourth type are the multifactorial, more polygenic disorders. I prefer the term multifactorial because it says what it means. It's many factors. So, in the chromosomal abnormalities, there is usually an abnormal number of chromosomes, or pieces of chromosomes are lost. There's a break, and substitution or something, that where the chromosome itself is in error, and the error happens during the crossing over and during meiosis and mitosis. It's actually quite common. 1 in 200 newborns has some chromosomal abnormality. Now, it may not express as much, or it could be a disorder that is not compatible with life. It kind of runs the whole gamut. So aneuploidy is when you have an abnormal number of chromosomes. So the abnormal number could be too many or not enough. And we look at the pairs. So monosomy is when you only have one of the pair, and that's usually not compatible with life, especially when it's with the autosomes, with the non-sex-linked ones. Polysomy is when you have 3 or more copies of the chromosome, and it's not a pair. This may be compatible, but usually there is a severe disability with it. So a common one, and that we can talk about, and that you will see, and you probably know people that have this, is trisomy 21, better known as Down syndrome. So the 21st chromosome, the 21st pair, there's actually 3. That's why it's trisomy. So one of the parents in meiosis 2, stays with 2 copies of one of the chromosomes. They don't separate out. Now, interestingly enough, that means that another gamete that person would produce would be missing chromosome 21, and so then that gamete wouldn't be viable. Down syndrome, trisomy 21, is the most common chromosomal disorder we see, 1 in 700 live births. Now, it's interesting, I learned this many years ago in a genetics course in the 70s at McMaster University. The Dr. Threichelt, who was the head of the genetics department, probably the whole biology department at McMaster, was my professor, and he stood up and said, these people have mental retardation, and they have all these features. And he was carrying on about it, and his hand goes up, and she said, Dr. Threichelt, I disagree with that. Turns out she ended up being the highest mark in the class, and she had Down syndrome. So mental retardation is not like 100%. But as a general thing, people have all of these issues. So a great many of them are delayed mentally. Uh, the tongue tends to be large for the mouth, so it often they walk and in a relaxed state, their tongue protrudes. Oftentimes, they're difficult to understand because they have trouble enunciating words because of the size of the tongue. Ears tend to be low set. They're lower in the temporal region, and so it looks slightly off. They look like the cranium is too large because the ears are too low set. They tend to have these little folds on the lateral aspect of the eye that are called epicanthic folds. Now, an epicanthic fold exists in other in other people. But we also see this as a trait of trisomy 21. They tend to be short and not have great muscle tone, and they often have heart issues, septal defects, and that kind of thing. They say increased susceptibility to respiratory infections and such. So, again, this is a generalization. Any individual with trisomy 21 may not have all of these characteristics, but they probably have a good chunk of them. For instance, not everybody with trisomy 21 have septal defects. Not everyone has poor muscle tone. But as a general list, this is what you see. Things like umbilical hernias. One of the things that I've seen a lot is this thing called the simian crease. When you look at the hand, for some reason, the lifeline and the, I don't know what the other line is, the headline, on most of us kind of run parallel to one another and curve. The one curves up towards the middle finger and the other one curves down towards the pisiform. Well, on people with trisomy 21, it tends to be one crease that goes straight across, and they call that the simian crease. I have met quite a few people. There's one patient comes to mind right now. She has an epicanthic fold, and she has low set ears, and she has a simian crease. But she is normal height and above average intelligence. She's she's a successful woman. And when I first met her, I thought, oh, okay. She's showing some signs, but not completely. And then I found out that her son has trisomy 21. So you wonder whether she has it and it's not expressed in her phenotype, but it got passed down to her son. I really don't know. It's just an interesting story. Now, there are lots of other polysomal disorders, but we'll stick with the trisomy 21. Now, the sex chromosome defects is when it happens in pair 23. So sometimes people So normally, if you're XX, you're female. If you're XY, you're male. But some people have two X's and a Y, so it'd be XXY. And this is Klinefelter's syndrome. So because of the Y chromosome, the person is male, but it's almost like their sex chromosomes are competing, so they have many female characteristics, and they don't generally make testosterone. So what ends up happening is there's atrophy of the testicles, and with that comes infertility. Growth hormone doesn't stop, so because they don't reach puberty, they keep on growing. So they tend to be very tall with long arms and long legs. They don't get male hair distribution, so they don't grow beards and that kind of thing. They often grow breasts, so it's gynomastia is that. They don't hit puberty, so voice doesn't lower. And there often is an impaired intelligence in these people. So this is Klinefelter's syndrome. And here's a picture of a person with Klinefelter's. You can see the gynomastia. You can see the female hair distribution. But this person has identified as male. Again, we have to be more careful around these sorts of things now than maybe 20 or 30 years ago when I was taking this stuff. It's how people identify, with the whole transgender things that we have to be sensitive to. There's something called Turner syndrome. Now, Turner syndrome is monosomy, but it's not of an autosome. It's of the sex chromosomes. So monosomy X is when one of the parents doesn't give a sex chromosome. If there's only a Y chromosome, monosomy Y, that's incompatible with life. But monosomy X is compatible. So because there's only an X chromosome, there's no Y chromosome, then this person is female. Now, usually the problem is that the father doesn't have a Y chromosome in the sperm, and it's often with elderly fathers. These people have high mortality rates postnatally. They tend to be short. They have fibrous ovaries. They're sterile. They amenorrhea, congenital heart defects, a lot of different characteristics. But they are female. Now, you'll notice how wide the neck is here on this person. Oftentimes, they kind of present, they look a little bit like somebody who has trisomy 21, but not when you look at all the characteristics. But I think that there's this genetic abnormality look that happens. Okay, the next thing we're going to talk about are the Mendelian gene disorders. So the single gene disorders. And we'll talk about this Punnett squares in a second. So there's a couple of definitions. Heterozygous and homozygous. So these two terms, heterozygous and homozygous, refer to the alleles in the two genes in the pair. So when the two alleles are different, we have a dominant and a recessive, then that gene is considered heterozygous. When the two alleles are the same, either both dominant or both recessive, that's considered homozygous. Now, genes, all the genes in our body, really what they're doing is they're a recipe, they're a code for making a specific protein. And those proteins may be structural proteins, they may be enzymes, they may be regulatory proteins, they do a lot of different things in physiology, but really its expression of genes is making a protein that causes either chemical reactions to happen or causes structure. And if there's an abnormality in the gene, in the recipe that the gene expresses, then it's going to be a protein that is slightly off, which may not act as an enzyme properly, or may not give the correct structure, or may not regulate what needs to be regulated. Now, most of these things are passed on, they're inherited, but as we said before, some of them could come from a mutation just that happens novelly in the fetus, and it's just a mistake, that a mutation that ends up being compatible with life and becomes a new trait. So when we look at the Punnett squares, if we have two heterozygous parents for allele A, the dominant is capital A and the recessive is small case, lowercase a. If there's two heterozygous parents, then what's going to happen is we'll end up with one homozygous dominant offspring, two heterozygous offspring, and one homozygous recessive offspring. Now, these are all probabilities. This is over a large sample. So say, for instance, the two parents are both carriers for the gene for polycystic kidney disease. Well, they probably don't know they're carriers for that, but one of their children, if they were to have 4 children, the probability of one of the children having not being a carrier is 25%. The probability that one of their children will be a carrier is 50%, and the probability that one of their children has polycystic kidney disease, autosomal recessive polycystic kidney disease, is 25%. Now, this couple may only have one child, This couple may have 6 children and none of them have the disorder, because we're only talking probabilities. It's only a 25% probability. When we look at the offspring of thousands of couples in this situation, that's when these probabilities start to really manifest and become more predictable. So there is this kind of gamble going on. Now, if one of the parents was heterozygous, up here, A, and this parent was homozygous, capital A, then we would get this one, capital A, this one, capital A, right? And so that would be 50% probability of a homozygous dominant, and this would be capital A, small a. This one would be capital A, small a. So it would be a 50% of the offspring would be heterozygous carriers. So this square is very useful, especially in genetic counseling, when these single gene traits appear. It helps people understand the odds of them having another child that has the same disorder. Now, in the not so distant past, the only way that you found out that you were a heterozygous carrier and that your partner was a heterozygous carrier was by having a homozygous recessive child with the disorder. And in that case, it's 100% of that child has the disorder. So this is really only useful for genetic counseling about making a decision whether to have another child. Now with genetic testing, we are much better at picking up, and then the counseling happens beforehand. We can know that things run in the families. Now, with that said, you're not going to be tested, not everyone in the population is going to be tested for every autosomal recessive disorder. That would be impossible. So we look at things where these disorders run in the families, and people get tested for specific things. Okay, so the autosomal dominant disorders are when the abnormal gene is dominant. It's the capital letter of the allele. When it's autosomal, that means it's the 22 chromosomes that aren't the sex-linked chromosomes. So because of that, men and women are equally affected. And one of the parents that's going to pass on that dominant disorder also has the disorder. Because it's dominant, unaffected people don't transmit the disorder. Now, if the person has the disorder and their partner doesn't have the disorder, there's a 50% chance that the child will have the disorder, because the parent that has the disorder was probably a heterozygous and can pass on the recessive gene. If both parents are affected, so they're both heterozygous with the effect, then 3 out of 4 of the offspring will be affected, and only one, there's only a 25% chance that they will pass on the 2 recessive normal alleles and the child not being affected. So An example of this is Marfan's. And Marfan syndrome, we could get into the biochemistry of it all, but we won't. They end up having a problem with the way they make collagen and its connective tissue. It becomes most noticeable in the osteoid, or the bones. So the long bones are extra long. These people are tall and slender. They have spidery fingers, and they tend to have problems in the epiphyses. So they have deformities at the joints. But because of this disorder of the way collagen is made, they have weak walls of the blood vessels, especially the aorta. They have problems with heart valves. They have problems with the chordae tendineae in the heart. So, but there are people out there that have this disorder that have no idea. They just are good at basketball, like they're tall and thin. When I was in chiropractic college, there was a guy in my class that had Marfan's, and his kid had Marfan's. So it didn't affect his cognition. It didn't affect anything except that he had these characteristic things. Now, this picture, this guy has these sternal deformities that may or may not be present. There's all kinds of things. Each individual is an individual. But that's the general idea. Huntington's disease, I've already made mention of this. It's a neurological problem, and it's called Huntington chorea. And what happens is proteins are made and they aggregate in the brain tissue, and it becomes a deterioration of the central nervous system, and it takes about 40 years of accumulation of this to start having profound effects. Now, chorea is spelled with a C, refers to dancing, and what happens is there's involuntary movement, and like this isn't just tremors. This is arm swinging and flailing and leg kicking, and it's quite pronounced. One of the problems with this disease is that people don't necessarily know they have it. So the most famous case of Huntington's disease was a folk singer. His name was Woody Guthrie. He wrote a song called "This Land Is Your Land" and various others. He was very big in protest songs and political songs of the late 50s and the early 60s. Well, he had Huntington's disease, and he died in the late 60s from it. But it was severely debilitating to him. And it got a lot of publicity because a lot of people had never heard of this disease before Woody Guthrie. Now, at about the time that Woody was being diagnosed with it, his son Arlo Guthrie, who wrote a whole bunch of great songs and was probably as famous or more famous than his father. He wrote a song called Alice's Restaurant and various other big hits of the 60s and the 70s and the 80s. He didn't know whether he was going to have Huntington's disease or not. And there were an awful lot of people like kind of waiting for him to turn 40 to find out whether he was going to go the way of his father. It turned out that he didn't. But interestingly enough, he decided that since it was a 50% possibility, then he was going to be very prolific, and he recorded an awful lot of albums before the age of 40 because of it. So it's an autosomal dominant, and because Woody Guthrie had it, there was a 50% chance that Arlo, his son, or Jody, his other son, who's also a musician, would have it. It turns out that neither of them do. They are alive and well all these years later. So the autosomal recessive disorders are much easier to pass on because parents are asymptomatic carriers. So in the autosomal dominant, the chance that you know that you have the disease is fairly high, and may not choose to procreate unless it comes on later, like Huntington's. So autosomal recessive is when it's on the autosome, so men and women are equal, and both parents have to be carriers if the trait's not expressed in them. They're heterozygous carriers. So if two carriers have a baby, there's a 25% chance that the disorder will be in the child. There's a 50% chance that their children will be carriers, and there'll be a 25% chance that the child will be a normal homozygous non-carrier. If one parent actually has the disease and the other parent is a carrier, then those numbers will correspondingly be higher. So it becomes 50% of the children will be homozygous with the disease, and 50% will be heterozygous carriers. Now, these autosomal recessive disorders are probably the main reason why we have cultural taboos about procreating with relatives, with siblings or with cousins, and the term for that is consanguinity. Sanguine is blood, so that means sharing blood. So when people are related, the chances that they have the same genes is increased, and so the chances that both parents are carriers can be higher, because carriers run in the family, as it were. We see this in problems in royal families. There are certain autosomal recessive disorders that kind of run in some of the royal families across Europe because of being closely related. So cystic fibrosis is a common one. We see it a fair amount. It's a single gene disorder. The problem is in a protein that's the transporter for chloride ions. Um, because of that, there's an imbalance of sodium chloride. It affects then osmosis. Because of that, the secretions from glandular tissue are too viscous. They're not watered down enough because it affects this chlorine affects osmosis. The places where it seems to manifest the most are in the pancreatic ducts and the bronchioles, and from that they have people with cystic fibrosis often have problems of the exocrine function of pancreas, not endocrine, and have problems with their lungs. Now, they also have very salty sweat, and that's an interesting finding. If you kiss a baby on the forehead and that baby has cystic fibrosis, they taste salty. Very interesting finding. The sex-linked disorders are disorders of recessive or dominant genes, but it's usually recessive genes that are found on the X chromosome, but there's no corresponding allele on the Y chromosome. And that's the problem. This doesn't happen with the Y chromosome because there are really no corresponding genes on the X that don't exist on the Y. So the only time we get Y chromosome disorders, they behave exactly like the autosomal disorders, because with a dominant and recessive allele on a gene that is on both X and Y chromosome. So it behaves exactly like an autosomal disorder. In order to be special, it's really X-linked. And it's recessive, right? So the problem here is that women can, because they have two alleles, they have two X chromosomes, can carry a dominant and a recessive and be carriers on the on that allele. They would be heterozygous. The problem is that their sons don't have, they get a Y chromosome from their father, so the mother is giving one of her X chromosomes to the son. If they are heterozygous, 50% of those X chromosomes will be carrying the recessive disorder. So that disorder will be expressed in the phenotype of half of their sons. So if the recessive allele is given to the son, then the son expresses it. If the recessive allele is given to a daughter, that daughter gets the dominant allele from the father and therefore is heterozygous and will not express the disorder. Women can get it and can get these disorders, but that means that the father must have the disorder and the mother is a carrier. So if the father has the disorder and the mother is not a carrier, is homozygous, then the father will give the recessive gene, the recessive X chromosome, to all of his daughters, but none of his sons. So unaffected men just don't carry the defective gene. So if a woman is the carrier, 50% of her sons will have the disease, the disorder. 50% of her daughters will be carriers of the disorder. If the only way that women can get the disorder is if the mother is affected, or if the mother is a carrier and the father is affected. I know that sounds all complicated, but if you work it out with the Punnett square, you'll see it. Hemophilia is hemophilia type A. Falls into this. It's a bleeding disorder. There's problems with different clotting factors, and they bleed easily from minor injuries. This is a sex-linked X chromosomal disorder, and it happens in male descendants of Queen Victoria. So the son of the Tsar of Russia had it. It runs in those royal families because of it. Another common one here is Duchenne's muscular dystrophy, which we've already talked about. So the multifactorial disorders, they run in families, very common, several genes and environment. So this is genetics loads the gun, environment pulls the trigger. They think cancer is this, congenital heart defects, various hypertensions, diabetes, type 1 diabetes especially, cleft lips, cleft palates. We can take this further, and if we drop the word disorders, but multifactorial characteristics, we can look at things like skin color and all kinds of things that are not disorders, but follow the same rules. Okay, so the congenital disorders that are environmentally induced, so they are not genetic, all revolve around these things called teratogens. These are factors in the environment that cause congenital malformation. Fetal vulnerability varies to these teratogens depending on what the fetus is doing at the time. So before the 3rd week of gestation, then it's going to go one of two ways. A teratogen will damage so many cells that the embryo cannot become a fetus, and the pregnancy terminates spontaneously. Or there's damage to a few cells, but not enough, and so it becomes insignificant. So there's not very much vulnerability before the 3rd week of gestation. Between weeks 3 and 9, however, this is when the organ systems start to develop. And we can get these congenital malformations because that's what the embryo is doing at that time. So they become very vulnerable, weeks 3 to 9, with weeks 4 and 5 being the peak susceptibility. Oftentimes, this is about the time when a mother finds out that she's pregnant, which can be fairly dangerous. After that first trimester, then during the fetal time, this is all just about growth and the maturation of organs, but the blueprint is already laid down. So the teratogens have very little effect at this point. It just affects growth and the way that things mature, but you don't get the malformations quite. You get maybe problems with function. So what do these teratogen agents do? They cause errors in the cells by screwing up the way they undergo mitosis, about the way that they differentiate. We talked about this way back at the beginning of the course, and cellular differentiation, and how environmental factors play a role on determining the differentiation of cells. That is, these agents, these things interfere with that. Now, when we're talking about all of this stuff, and we have a vested interest in the health of our children, we start to look at things about diagnostic tests versus diagnostic screening and the differences between them. This is really a topic for the discussion board, and I want you to think about this, and I'm looking forward to seeing what people have to say and what they discover about screening versus testing, and the populations involved. So, as I said before, the teratogens have their effect on the development of organs, and the embryo is at risk when that organ is being, or that organ system is being developed, and that's when they're vulnerable to those teratogens. So if we kind of look at the at when the organ systems develop in gestation, we can see what organs are going to be vulnerable in what week of embryonic development. So when we look here, the central nervous system is a very early system to develop, and it basically gets laid down between the beginning of week 3 and middle of week 6. So that's when the nervous system is vulnerable to a teratogen. If the exposure to the teratogen happens in week 7, it's probably not going to affect the central nervous system of the heart, but maybe problems with the limbs, eye development, genitalia. There was a drug a few years ago, quite a few years ago now, called thalidomide, and it was given to pregnant women as an anti-nausea drug to combat morning sickness. And it turns out it had it was a teratogen. But it was kind of given in the second to third month of gestation, because that's when the morning sickness started to manifest. So these children were born without arms or without legs or really malformed genitalia and things like that. And they were called thalidomide babies because everything, heart and central nervous system and that, were all working fine because the exposure to the teratogen came slightly later. Right. Anything that happens before the development of central nervous system and heart probably is incompatible with life. And if it happens kind of after week 9 or thereabouts, you're only going to get morphological defects, not really developmental defects. So there's a couple of different types of teratogenic agents: chemicals and drugs, alcohol, recreational drugs. Sorry about the spelling for fetal alcohol here. That's getting a lot of press. Women that drink during, smoking cigarettes becomes a big thing. So these are teratogenic agents that fall into this category. Viruses are really important, and it's one of the reasons why women have to be very careful around red measles, rubella, the cytomegalovirus, herpes. So the important viruses have the acronym TORCH, toxoplasmosis, others, rubella, cytomegalovirus, and herpes. Generally, others would be at the end, but these are the things that are dangerous. And radiation is a problem. And this is why we don't x-ray pregnant women. You have to be careful about dental x-rays. You have to be careful about bone x-rays. Less so, I mean, this was a bigger problem with old cathode ray TVs. They put off some radiation and things like that. But the big thing is x-rays. And it's the reason why when you go to the dentist to get dental x-rays, they put that big heavy lead apron on you, especially to cover your lap. Yeah. So any one of the viruses can lead to microcephaly, heart defects, liver defects, spleen defects, bruising, petechia, that kind of thing. But these are very real issues during pregnancy. Very good reason to be immunized against these viruses if you're planning on having children. So that's it for this unit.